Hi, this is Colleen Belk. I'm one of the authors of your textbook, and I'm going to try to help you understand a process called mitosis, where the nucleus of a cell divides in order to produce from one parent cell two identical daughter cells. So we'll start with one parent cell, and we'll end with two daughter cells that are genetically identical. So this happens during the division of the nucleus called mitosis. Preceding mitosis is a part of the cell cycle called interphase, and then after mitosis is cytokinesis, which is division of the cytoplasm. Mitosis itself consists of four stages: prophase, metaphase, anaphase, and telophase. And before we describe what goes on in each of those stages, we might want to understand why cells undergo cell division. So cells undergo mitosis in order to reproduce. Some cells reproduce asexually by mitosis. Cells undergo mitosis to grow. If an organism needs to get bigger, it can undergo mitosis. And cells also undergo mitosis in order to repair wounds. So for instance, if you cut yourself, your skin cells will undergo mitosis to produce new healthy daughter cells. So during the first stage of mitosis, prophase, we can see in this nucleus that has 4 chromosomes that the chromosomes are now duplicated X-shaped chromosomes. So by the time we get to prophase of mitosis, the chromosomes have been duplicated. That happened during interphase, and they're connected to each other at the centromere. So the duplicated chromosomes are each composed of a pair of sister chromatids. So this cell has 4 chromosomes and 8 sister chromatids in its nucleus. Sister chromatids are identical copies of each other. Cells make these identical copies so that they can separate them into daughter cells. So it's almost like having 4 documents that have been duplicated and attached with a staple at the center. So at prophase, we have duplicated chromosomes attached at the centromere, and also we have the formations of some microtubules, and these are protein structures that kind of act like lassos in order to move chromosomes around in the cell. At metaphase, the chromosomes align across the middle of the cell. So this cell with just 4 duplicated chromosomes, they line straight across the equator or the middle of that cell. And we can see that there are microtubules, and these microtubules help position the chromosomes on the equator of the cell. At anaphase, the chromosomes separate from each other, the sister chromatids pull apart, and now they become individual chromosomes again. So at anaphase, the sister chromatids pull apart to the poles of the cell, and then each one becomes a chromosome again. So in this whole cell, there are 8 chromosomes now, because you're only a sister chromatid when you're attached to each other at the centromere. This particular chromosome is identical to this one. Likewise, this chromosome is identical to this one, and so forth. So the microtubules are retracting and pulling the chromosomes to the poles of the cell during anaphase. And then at telophase of the cell cycle, the nuclear envelope is going to reform around our separated chromosomes, and we also see the beginnings of what's called cytokinesis. So the microtubules now form kind of a belt that wraps around the cell and pinches it in two. So cytokinesis, or division of the cytoplasm, occurs on the heels of telophase when we have our separate daughter nuclei and separates that one large cell into 2 separate daughter cells. So the result of this then is we have 2 daughter cells that are identical to each other and to the parent cell. So in summary, mitosis is a type of cell division that produces 2 daughter cells that are genetically identical to each other and to the original parent cell.